Will give a seminar on the Watson and Crick model of DNA in the subject of molecular biology. Friends, in this Watson and Crick model of DNA, first I tell basics to you. In the PPT, the first page of heading is Watson and Crick model of DNA, some basic importance. Nucleoside is a compound formed by the combination of a pentose sugar and a nitrogen base. Nucleotide is a compound formed by a combination of nucleoside and a phosphate group. Nucleotides building blocks of nucleic acid. Nucleotide have three characteristic components. What's that? They are a nitrogenous base, a pentose sugar, a phosphate group. In this subgroup, nitrogen bases are present in DNA. One is adenine, cytosine, guanine, thyrosine. The different types of nucleosides of DNA is 4. First, deoxyadenosine. Second one, deoxyguanosine. Third one, deoxycytidine. Fourth one, deoxythymidine. Okay, the DNA contain two purine bases. One is adenine, second one is guanine. The DNA contain two pyrimidine bases. One is cytosine, second one is thymine. So the third page, the fourth page, we will see the structures of the purines and pyrimidines. We will see, friends, in the fourth page of the PPT, we will observe the purines and the pyrimidines. Okay. Next one. Fifth page also we will see the nucleotide symbols and a nucleoside. What they are having in the picture is dedicated to the structures. Okay, we will next to the tabular form of the nucleotide and nucleic acid nomenclature. We will observe in the PPT 7th page. First, the column is base, nucleoside, nucleotide, nucleic acid. First, purines, adenine nucleoside, adenosine, deoxyadenosine. Adenine nucleotide, adenylate, deoxyadenylate. Nucleic acid, RNA, DNA. So the guanine, next base, guanosine, deoxyguanosine. Nucleotide, guanylate, deoxyguanylate. Nucleic acid, RNA, DNA. Pyrimidines, cytosine, cytidine, deoxycytidine, cytidate, deoxycytidate, DNA, RNA, nucleic acid. So that's the tabular form of the nucleotide and nucleic acid nomenclature. Okay. Next, the 8th page. The phosphodigester linkages in the covalent backbone of DNA. Here's the friends, the structure. Phosphodigester linkages in the covalent backbone of DNA. Okay, friends. The 9th page also, structures of the hydrogen bonding Watson and Crick. Here's the the blue lines. Blue lines are the hydrogen bonding Watson and Crick. Behind the H bond and the oxygen bond. Behind the H bond and the between. Sorry friends. Between the H bond and the nitrogen bond. Between the H bond, hydrogen bond and the oxygen bond. Between the oxygen bond and the hydrogen bond. In the guanine, adenine. Between the hydrogen bond and the oxygen bond. Nitrogen bond and the hydrogen bond. In adenine, this is the hydrogen bonding Watson and Crick, friends. We will observe in the picture. And the 9th page we will observe A form, B form, Z form. This is the Watson and Crick model of DNA picturization. We will draw in the exam or the matter. Here's the information is A form, B form. That's the drawn pictures was given the information of the tabular form in the 10th page, friends. X-ray diffraction. X-ray crystallography was originally used to look at the structures of the simple organic minerals, but also progressively applied to more and more complex molecules. It aided in the determining of the structures of the alpha helix and the beta sheets and the hemoglobin and DNA. X-ray diffraction photo, friends. X-ray diffraction of DNA. Okay. And the 14th page is DNA is double helix. Friends, in the Watson and Crick model of DNA, the DNA has double helix. To shed more light on the structures of the DNA, Rosenhild and Franklin and Winkins used the powerful method of X-ray diffraction of the analysis of DNA fibers. They showed in the early 1915s the DNA produced a characteristic X-ray diffraction pattern. From this pattern, it was deduced that DNA molecules are helical with the two periodicites along their long axis. The primary one is 3,4A and secondary one is 3,4A. X-ray diffraction is the structure of DNA. Watson has shown in the picture by the Wilkins in early 1953. From the picture is possible to calculate the distance between base 3.4A, the length of the period 34A, the rise of the helix 36 degrees. This is the picturization of the Watson and Crick model of DNA. Watson and Crick model of DNA. In 1953, Watson and Crick proposed a three-dimensional model of structure. It consists of two helical DNA chains wound around the same axis to form a right and handed double helix. The hydrophilic backbone of alternating deoxyribose and phosphate groups on the outside of the double helix. The purine and the pyrimidine bases of both strands are stacked inside the double helix. Okay. The steps are formed by the paired nitrogen bases. A always pairs with the T with 2 hydrogen bonds and the G pairs of C with the 3 hydrogen bonds. Hence, true strands are complementary to each other. Because of the speciality in the base pairing, the amount of the purines is equal to the amount of the pyrimidines. A is equal to T and G is equal to C. Here's that this is called Chargaff's rule of base equivalence. Chargaff rule of base equivalence. The DNA double helix is held together by two forces and hydrogen bonding between the complementary base pairs and base stacking interactions. These two forces gives the stability of double helix. Friends, we will remind the Chargaff rule of base equivalence is including in the Watson and Crick model of DNA. Okay friends, the discovery of the structure of DNA by Watson and Crick here's the held in year 1953. It was a momentous event in science. A event gave rise to entirely new disciplines and they influenced the course of many established ones. Okay friends, thank you. For more videos, subscribe the channel.